Hello, everyone. My name is Amala Salif. I'm going to be discussing about the functions of nucleic acid in the cell. Uh, First, we have to talk about, like I said, my name is Amala Salif. This course is biochemistry. My presenter knows introduce nucleic acid as essential biomolecules responsible for storing, transmitting, expressing genetic information in all living organisms. What are nucleic acids? Nucleic acids are biological macromolecules composed of nucleotides monomers. There are two types of nucleic acid. They are the deoxyribonucleic acid, which we call DNA, and the ribonucleic acid, which we call RNA. They are essential for hereditary and protein synthesis, which includes the ribosome in the cell. They are found in all living cells. The idea is to explain that nucleic acids carry the instructions necessary for cellular structure, function, and reproduction. The next slide, I'm going to be talking about the structure of nucleotides and what these nucleotides contain. Nucleotides contain a phosphate group, a 5 carbon sugar called pentose, a nitrogenous base. And for the DNA, which is one part of nucleic acid, the DNA has the bases, and the DNA bases contain the adenine, thymine, cytosine, and guanine, whereas RNA bases contain adenine, uracil, cytosine, and guanine. When you look, you know that thymine is replaced by uracil in RNA. The note for this discuss how nucleotides join through phosphate diester bonds to form nucleic acid chains. The next slide, I'll be talking about DNA has the cell information storage system, how it contains information about the cell. DNA stores hereditary information, that is the transmission of traits from parents to offspring. They're located primarily in the nucleus of the eukaryotic cell, which is the nucleus is the one that controls the cell, is the center portion of the cell where all cell activities occur. It contains genes that determine traits and cellular functions. It also replicates before cell division to ensure genetic continuity. My notes here is to compare DNA to a biological instruction manual that contains the code of life. Next slide, we're going to talk about DNA replication. Replication is the process in which both living brings some of its own kind, exactly looking the same. So DNA replication copies genetic information before cell division. It ensures each daughter cell through mitosis cell receives identical genetic material. Semi-conservative process involve complementary base pairing. An idea here is to emphasize the importance of accurate DNA replication for growth and repair. Next slide talk about RNA and gene expression. RNA helps convert genetic information into proteins. There are three major forms: the messenger RNA, transfer RNA, and ribosomal RNA. They play a very critical role in protein synthesis, especially the ribosome is the one that synthesizes the protein, whereas the Golgi apparatus package the protein. The idea here is to explain that RNA serves as the intermediary between DNA and proteins. Next slide, we'll talk about transcription of DNA to RNA. First step of gene expression. Then RNA polymerase synthesizes messenger RNA from DNA template. This occurs in the nucleus of eukaryotic cell. An idea of my node is to describe how specific genes are copied into messenger RNA molecules for protein production. Next slide is translation of RNA to proteins. Ribosomes read messenger RNA codons, then transfer RNA delivers amino acid. Proteins are assembled according to genetic instruction. So proteins are complex chain of amino acids that is very essential in cell structure and function. The idea is to explain how the genetic code directs the sequence of amino acid in proteins. The next slide we'll be talking about functional roles of proteins produced from nucleic acid information. 1. The enzymes catalyze reactions. Structural proteins support cells. We'll also look at the transportation of proteins that move on molecules, and regulatory proteins control cellular activities in the cell. Our presenter notes of the idea is to connect nucleic acid function to production of proteins that perform most cellular tasks. Next slide, we talk about the regulatory functions of RNA. MicroRNAs regulate gene expression. Small interfering RNA can silence gene. RNA influences development and disease processes. The idea is to discuss how RNA molecules regulate regulate which genes are turned on and off. The next slide we'll be talking about nucleic acids and cell division. DNA replication precedes cell division. Genetic information is transmitted to daughter cells, essential for growth, repair, and reproduction. The idea highlights the importance of nucleic acid in the maintaining of genetic continuity. Next slide, we'll talk about nucleic acids in biotechnology and medicine, DNA sequencing, genetic engineering, gene therapy, and forensic analysis. Personalized medicine. Ideas to discuss modern applications of nucleic acid research in science and healthcare. And the conclusion of this presentation comes to the fact where DNA and RNA are fundamental to life, which of course we know they contain the code of life. DNA stores information, whereas RNA expresses that information. And together they control growth, development, reproduction, and cellular function. Understanding nucleic acids is essential to biochemistry and modern biology. The idea is to reinforce the concept that nucleic acids serve as the molecular foundation of life. And my research or references or scholarly references can be seen from the following scholars: Albert B. Johnson, Bruce J. M., and Nelson D. L. and Cox, Legionnaire Principles of Biochemistry, and UALA King. Thank you very much, and this is my presentation. Thank you.